if you're talking about a permanent treatment for a blood disease, you have to be able to edit these stem cells specifically, because that way, decades and decades later, in a human, or months later in a mouse, all of the cells that the levels of correction will be maintained, and all of their blood cells will remain corrected. You have to hit the root, essentially, of the tree. So we have a protein and RNA complex together, along with a short piece of DNA. The protein, guided by the RNA, makes the cut in the genome, and then the DNA essentially pastes in. A crucial question is whether or not this gene editing treatment is lasting. We edit the cells today. Will the edited cells be around to keep you healthy years from now, potentially? So to test that, we edited human cells in the laboratory. We then injected them into mice. And then we looked at the editing of the cells 4 months later and asked whether or not the edited cells hung around. When we do that, we find that about 2% of the stem cells that remain in the bone marrow after 4 months are edited, which in the case of sickle cell disease is that level of editing is likely to have clinical benefit, but we would like to get a little bit better than 2% in the future. It'll be quite a while before we're confident enough in the safety of CRISPR-based procedures to move into the clinic. And in the case of this project specifically, it'll be years of very careful and rigorous study to ensure that it's totally and completely safe before we even contemplate an actual clinical trial. But we have a path. We just have to walk down it.